Hello, my name is Angel Estrada and I'm going to present my work title, How does the candy barrel cactus Echinocactus platyacanthus do to resist dry season? Introduction. Arid zones are characterized by marked dry seasons that generate weather stress. Plants such as Echinocactus platyacanthus, a large and long-leaf globose cactus from Mexico, show high resistance to such conditions, but the mechanisms that allow it to do so are unknown. To understand better this process, was proposed to identify the possible physiological mechanisms that have allowed Echinocactus platyacanthus to survive and grow during dry season in an arid zone, and if this interaction with the rhizospheric bacterial community has influence in those mechanisms. Material and methods. The sampling site was in Guadalcázar, San Luis Potosí, Mexico, outside of Chihuahua Desert. 10 adult plants of Echinocactus platyacanthus were selected. The sampling was carried out in the rainy season, September, and another in the dry season, March. Sampling from the plant tissues, rhizospheric and bulk soil, were taken for further analysis. Indicators of physiological stress were evaluated, such as chlorophyll fluorescence to evaluate the conversion of light to chemical energy. Tissue acidity signs. These plants absorb carbon dioxide during the night and accumulate it as malic acid. Pigments, chlorophylls, and carotenoids are essential for capturing light and protecting against high radiation. The water potential of tissues was evaluated to find out if the plants lost water. Reactive oxygen species were determined by the production of the hydrogen peroxide. It is usually produced due to stress and harmful to the plant. And the production of root exudates focusing on the amino acid to see their relationship with abundance and recruitment of bacteria. Rhizospheric and bulk soil samples were taken to evaluate the composition of the bacterial community. In the laboratory, DNA extraction and sequencing of the 16S rRNA gene by Illumina MySQ were performed. This data was analyzed with motor program and the sequences were classified with the Silva database. Differences in the bacterial community composition were evaluated by multivariate analysis. The number of copies of NIF-H gene for nitrogen-fixing bacteria and 4D for phosphorus solubilizing bacteria were also quantified. Results. During dry season, the concentration of oxygen species increased, which possibly damaged the chlorophyll since their concentration decreased. However, the plant response increasing the amount of carotenoids has protective compounds against oxidative stress. The water content decreased, possibly reducing the absorption of carbon dioxide, which was reflected in the decrease in the nighttime acidity. The amino acid exuded by the roots increased, for example, serine, glutamic, methionine, glycine, with the drought, and could have a direct relationship in the chains of the bacterial community. Entotenalase, actinobacteria, Pyrrhula, Nehalococcus, Hematase were the most abundant genera in the dry season. They had been generally reported to have the decomposition of organic matter, humus formation, nitrate formation, and enzymatic activity to relate to the phosphorus disposal. Also, nitrogen fixers increase during dry season, possibly because in this condition nitrogen is more limited. All these mechanisms together may be contributing to the plant's tolerance to stress, which has allowed it to survive in arid environments.